Hi, I'm Zwijsen. I'm a basic scientist at the biomedical faculty of the University of Leuven, KU Leuven, and I'm a vessel biology specialist. Okay, what are the questions that we address? Well, we study lymphatic vessels. They're also called the milky vessels because the fluid that the lymphatic vessels drain comes from our tissues and is kind of whitish. And it's that white color that gives the name to these vessels, the milky vessels. So perhaps you know someone, or maybe you are someone that lives day to day with lymphedema. And lymphedema is the swelling of tissues due to an impaired lymphatic system. And that causes really painful tissue swellings that are causing a lot of discomfort and heaviness. Up to today, there's no real cure for lymphedema, just the lifelong management of the disease symptoms. So by studying how and when lymphatic vessels form and function, we get a deeper insight in mechanisms that may lead to improved treatment of lymphedema. What methods do we use? Well, we use human cells. Human cells that we can use to build rudimentary lymphatic vessels in the lab. These we call vessels on a chip. And these help us answering relatively straightforward questions that involve just a few different cell types. However, some questions we cannot study in a dish in a lab. For example, lymphatic vessels, they form mini valves that function as one-way doors that steer the flow direction into one direction and prevent backflow. Well, these mini valves we do not manage to form in our vessel on the chip. How comes? Well, we still miss and we still do not fully understand all the cues that control when, where, how, and how many valves it will form. And that's then when we turn towards the mice. So we study lymphatics also in the mouse using very early stages of normal and disease development of the vessels. Results from these studies fuel new hypotheses that are then used to test in the vessel on a chip, to improve the vessel on the chip, and vice versa. So these type of experiments really go hand in hand, and combining both approaches delivers different pieces of one bigger complex jigsaw, which helps closing really relevant knowledge gaps. Why do I value openness on laboratory animal research? Well, over the years, I have been talking a lot with my friends and family and students as well, other scientists, about animal research. And I was always amazed that many amongst us actually do not realize on how animal research has and is shaping our daily lives. In the medical world, think of the painkiller you took yesterday, the contraception, IVF, the medical device, the pacemaker that your father carries, and so on and so forth. All in each of these developments have reached the clinic based on animal research. Why do we not know about this? Well, many scientists feel uncomfortable and sometimes a bit unsafe to speak up about how they perform animal research and why. And events like today, like the Board 26, are really great. You probably all have heard about the strict and progressive EU legislation. And what matters really a lot to me is also that, just like in the hospital, performing animal research is not done in isolation. So it's really a joint responsibility of everyone involved. Together, we do our best to ensure both excellent animal care and high quality research, and being open about it and exchanging experiences to further improve this care day by day is really key. So thank you very much and happy continuation of board 26.